everyone, welcome to another lecture of drug delivery engineering and principles. We are at gene delivery module and we are discussing about various aspects of gene therapy either by cells or without. So, let us quickly do a recap of what we learned so far. So, as I said, we are talking about gene therapy. So, the first thing we talked about is for gene therapy is let us deliver a cell that carries the gene of interest. So, this could be a native cell itself that is expressing that gene and contains that gene, or this could be a cell that you have engineered first in laboratory to express this gene of interest and then deliver it. So, that is one option, and that is very similar to what we have discussed earlier in the previous classes, where you can encapsulate this cell in a semi permeable membrane that will prevent any kind of immune response from acting in, but the cells can continue to express and deliver the protein of interest. The problem with that is it is not fairly successful, because there are several small components also present in the immune system that will still be able to go through this membrane such as cytokines and complement, and they will not let these cells survive. So, that is one challenge. And so, what is seen, and we discussed an example in a mouse setting, that if you have a immunodeficient mouse, these things survive at least for a few months, and but if you have a healthy functioning immune system carrying mouse, then those things do not really survive. They within a month or so we saw that mouse rejected the implant. So, that is a problem. So, that is where the second strategy would come in, is let us not talk about non-autologous cell. So, any cell that is not from your own body, use the cells that are in your own body. So, why can't we just deliver the gene? Hopefully, the cell will go or the gene will go to the cell that you want it to express, and that way the immune system will not really attack the cell as much. So, in that regards, we discussed several barriers that are present. Of course, it is all well and good to say that you can deliver a DNA, but then the delivery of the DNA has several challenges, because first of all, when you deliver this, there are several extracellular barriers. So, your serum contains lots of enzymes that can degrade it. It is hard for this to target the organ that you want. So, let us say if you want the expression to be in lungs, it is difficult that all the DNA will actually go to the lung. And then the immune system again will clear out quite a bit of it. Immune system, or you can also call it the reticular system. So, all of this will clear out quite a bit of it, and then there are several intracellular barriers again. So, even if it reaches your target cell, how does it go inside the cell? So, let us say if this is a cell. So, first of all, you will have to figure out how does your DNA pass the cell membrane, because DNA is a fairly charged and large molecule. So, it cannot go through, so it has to go through endocytosis. So, if it goes through endocytosis, one of the problem is that lot of it will go to lysosomes and get degraded. So, that is one barrier. Another barrier is how do you, if you want to prevent that, how do you burst throughout this particular endosome to come out in the cytoplasm, and once it is out in the cytoplasm, how does it able to move from the cytoplasm from its location all the way to the nucleus, because again it is a large molecule, so its diffusion is fairly limited, and then once it reaches the nucleus membrane, how does it go beyond that, how does it go inside that, because again the same barrier is present on the nuclear membrane also. So, several barriers that are present. Nonetheless, it is not as bleak as it seems to be. People use it all the time, and at least from the literature reports and from the experiment, it does seem that you can get gene delivery into a cell despite all these barriers, and that is what we are going to discuss today. So, let us understand some of the barriers. So, let us talk about intracellular barrier, the movement itself. And so, again you can characterize the root mean square speed here, velocity here, where you are trying to figure out how quickly the DNA will move in there. So, it will depend on the diffusion coefficient of the DNA in that environment. And again you see that there is quite a lot of variation. So, you can see the MSDs can vary quite a lot, depending on the different condition used. So, if it is slow, it is like subdiffusive, whereas if the slope is higher, then you are talking about quite a bit indicates an active transport. So, if you have somewhat an active transport, you see that the MSD, mean square displacement, is almost. So, let us say let us take at the time 1 second. If somehow we can get an active transport, so we are looking at 10 to the power minus 1 micron square here and 10 to the power minus 4 micron square here. So, the diffusion is actually increased, the movement is actually increased by 3 orders of magnitude, which is a huge improvement in translocation. So, somehow if you can get the active transport to work, you have much better chance of getting your cargo into the nucleus. So, that is what these authors have tried. So, what they have done is they have designed a plasmid that has a binding site as a nuclear localization signal. And so, what this is, this NLS binds to a protein that can signal to the cell. So, let us say if a protein gets bound to this when bound to this DNA will signal the cell to move it to the nucleus. So, now you are actually relying on active transport once you do that. So, you put this nuclear localization signal and then the active transport will help it at least overcome one of the barrier, that is movement first of all into the through the cytoplasm. So, let us say if this is a big cell, here is your nucleus, here is your DNA. So, all the way from here to here, this NLS first of all will take care of this movement as well as movement across the nucleus membrane. So, so that is what it is being shown here. So, if you have different conditions, if you only have the plasmid, you do not really see quite a lot of production of the proteins. So, in this case the protein that is being used is something that is a luciferase gene. So, it can be easily measured using light production, and so you see and again this is on log scale. So, you see you can change the type of plasmid you are using, the conformation of the plasmid, you do not really get quite a lot of enhancement, but the moment you put an NLS you actually see quite a big jump in the localization or essentially actually production. So, this is more a functional assay. So, in the production of the protein through this. So, so that is one, okay, so that is good, we can overcome one of the barrier. What about the rest of the barrier? What about going into the cell? What about coming out from the endosome? And what about the extracellular barriers, the proteases, the nucleases present in the environment? How do you target a particular organ? How do you target a particular cell? So, for that we need a carrier. So, it is fairly clear that because of so many barriers, if you just inject the DNA by itself, it is probably not going to be able to reach in sufficient amount to your target site. Now that we established that yes, we need a carrier, we have two choices. One is we go to the nature itself and use a virus based carrier, and this is nothing but a virus that is already adapted itself to infect human beings. So, it has overcome all those challenges that we talked about. So, it has a shell of protein shell which protects it from any extracellular barrier. It is a different size now, so it can translocate, it has receptors to target a particular type of cell and organ, and that can be then used to deliver your DNA. So, you can take this virus, remove the viral genome, put your gene of interest into the virus, and then let the virus go and deliver your gene of interest to your target cell. And this is actually very, very efficient. and the whole reason is that they are well adapted to do this. So, this technology that is being made by evolution is very efficient, but the problem is of course that and we will discuss this in more detail, but obviously let us say if you are not able to remove all the viral genome, you are talking about causing now a new disease. The other option is to use polymers and lipids. So, what we were discussing throughout the course, so non-viral vectors, so some kind of material and mimic some of these viral strategies, either their size, put some targeting ligands onto this, change their charge on the basis of that, get different size particles and then package these particles with the DNA and then deliver it. So, these are two major sort of carriers that we can use for this application. So, let us look at the state of the current science with both viral and non-viral vectors. So, obviously viral vectors, as I said, has lots of pros. It is a very high transfection efficiency. So, you are talking about pretty much more than 50% efficiency. So, almost more than half of your cargo will get transfected and result in functional protein being produced. So, it has natural tropism, which means that there are viruses existing that infect only a particular type of cells and then there are viruses existing that infects many cells and then also being selective in the type of cells that they are infecting. So, that way you can choose what virus infects your target cell and use that. So, you do not really have to do much work, the nature has already produced all this to help you do it. Then these viruses have actually evolved mechanisms for endosomal escape. So, they are very well versed in how to rush out from the endosome and go back into the cytoplasm, because eventually these viruses also want to inject their DNA and want the DNA or the genetic material to go in the nucleus. So, they can do that. In fact, one of the strategies that we talked about, the proton sponge effect, was something that we learned from these viruses. So, in this case their coat protein was such that was acting as a proton sponge, and then they also have mechanisms for transportation of the DNA into the nucleus. So, again they are well adapted in overcoming all these challenges, and that is why they are one of the most ideal systems when you consider their efficiency in gene transfection. However, there are some cons, and one con again being that now that these viruses are not something that is a part of your own body. So, now you have introduced a foreign material, so your immune system will start acting against it. So, you get a very strong immune response against these viral proteins, and that means that let us say if you have done it once and you want to do it again, and by the time you are ready to do it again, the body is already aware of how to handle that particular virus, because you essentially vaccinated the body by giving the first dose, and the second time you do it, the body is going to clear it even before it can do any of its function. So, it prohibits multiple administration. Now, the problem is that these viruses, even though so one of the things that these viruses will do is, remember this chromosome that we had discussed, and let us say this is the gene that we want to put, but these viruses again are not very specific in where this gene is going to go ahead and insert itself. So, it could happen that this gene will insert on this chromosome, it could happen that this gene may go and insert in a neighboring chromosome, let us say here. So, on the basis of where it is getting integrated, this may lead to several consequences. First of all, the expression may change. The second problem is, what if now your gene, you have a promoter in a gene as well, so that it can express what it is carrying. What if downstream you have some oncogene? So, now along with your own protein, you are also making the cell cancerous. So, that is a big problem. Actually, there was a lot of enthusiasm for these viral vectors, and they went even in human clinical trials, but in one of those trials what they found out is more than 50% of the patient actually developing cancers, and the patient actually died because of that. So, that decreased the enthusiasm on using these viral vector. Of course, this was quite a long time ago and technology has since improved quite a lot, but there is still a concern as to something like this could happen and lead to more serious disease than what you are trying to cure. It is a fairly complicated synthesis process. It is not trivial to take a virus and then load it with your own DNA and let it go and do whatever you want it to do. It also has a limitation on the gene size that you can put. So, this virus code protein that you may have, it may only be of a certain size and there is a limit. So, let us say the virus genome is about 10 kilo base pair, then you can't really load more than 20 or so. I mean, it may have some stretchability, so you can add maybe few kilo base pairs, but eventually you can't really put 100 kilo base pair in there. It's just too big for this to be loaded into that space that is given in the viral shell. So, if your gene is big, you have to look for some other way. And again, there is toxicity associated with this because of the immune system. There is also a chance that you may contaminate your thing with a live virus, and if you do that, you again cause a different disease than what you are trying to cure, and that is never good. So, there are few cons that exist with these viral based vectors, and so that is why the field has now also tried to look into other alternatives to what these viral vectors offer. And so, one again, as we discussed, are the non-viral vectors. These are these polymers and lipid based vectors. And one of the major advantages is, unlike viruses, they are fairly low immunogenicity. We have seen this throughout this course that these polymers and lipids, there are so large libraries of it that you can choose fairly low immunogenic polymers and lipid. They will not typically develop an immune response against these proteins or these polymers and lipids. There is a big library, so you can use non-toxic biomaterial. They are fairly easy to synthesize, so we know how to synthesize this very well. There is all kinds of chemistries and you can do a very easy mass production. So, and that is a big plus, because if you want to, let us say, use it in clinics and you expect to get 1000 patients walking in every day in a clinical setting, you want quite a lot of this polymer and you can get that. You can potentially target them. So, you can, once you form the particle, you can conjugate it with targeting ligands. And that would be enough to help with some targeting. There is no really limit to the plasmid size. You can get as much plasmid size as you want. So, if you want 10 kilo base pair, that is feasible. You want 100 kilo base pair, that is feasible. So, large genes are easy to take it through, unlike the viral vectors where we were saying there is a limitation to the gene size and they do not really integrate. So, in this case, if let us say this is the nucleus and these are your chromosome. So, the viral vectors will not integrate, they will just lie separately and will produce gene from this separate plasmid. So, you do not have to worry about oncogenes being activated and other things like that. So, they can be administered fairly without concern of causing cancer and other diseases. However, like the viral vector, there are some cons associated with these systems as well, and let us look at what some of these cons are. So, the biggest con is it is a fairly low transfection efficacy. So, now here you are talking about more than 50% of the cargo transfecting. Here the efficiency is actually very low. We have been trying to improve that, but we have not become as good as the nature is in increasing this efficiency. So, this could be as low as 1% actually, it just depends on what you are using, and that means that almost 99% or more of your drug is getting inactivated or is wasted. most of the vehicles that we use are fairly toxic at high doses, and the reason we need high doses is goes back to the transfection efficiency. Now that because we have such a low transfection efficiency, we want to deliver more of it, so that at least even if 1% goes, we have enough dose. But then at those high doses you can have toxicity associated with these vesicles and with these non-viral vectors, and then there is no natural tropism. So you have to worry about how do you target a particular cell, how do you target different cells. Endosomal escapes become a challenge. So we said we adopted some of the proton sponge effect, but obviously we haven't come to the technology which is as good as the viruses in proton sponge. And then also the nuclear transport mechanism became an issue, because earlier we were talking about a DNA molecule having problems diffusing in the cytoplasm. Now we are saying that this DNA molecule is in a large particle, so it is even larger now. How this is going to diffuse in the cytoplasm is another bigger challenge. So all of these combined result in quite a lot of pros and cons for each of these strategies, and depending on what is it that is your application and concern, you choose one or the other. Obviously this is a drug delivery course and we have been talking about polymers, so we are going to focus on non-viral vectors. The viral vectors for gene delivery are still being used in research, and they have gone to clinics in the past as well. So this is another field, but we are not going to talk about the viral vectors much, we will focus on the non-viral vectors. Okay, so gene delivery where and how. So let us look at macroscopically what is happening. So you go to a clinic, the doctor injects you something. So these could be injections, these could be nasal spray, orally you take something, they put it under the skin, muscle, whatever. Then if you zoom into it as to what is happening there, you have some particles, you use either some targeting, or these particles inherently go into the cell, they go into the endosomes, and eventually if you have used proton sponge effect or something else, they burst this endosome and come out into the cytoplasm. Once they come out in the cytoplasm, typically the best strategy is to let your DNA come out, because this particle, as I said in the previous slide, is too big. Now we are talking about 100 nanometer, earlier the DNA was maybe 1 to 10 nanometer, now you are talking about 100 nanometer big particle moving in the cytoplasm, that is going to be a big challenge. So most vectors you can design in such a way that once they come out from the endosome, the DNA can release, and we'll talk about how this release can happen, but we expect the carriers to release in cytoplasm. And then either using NLS or some other strategy, we hope that this DNA will translocate to the nuclear membrane and go into the nuclear membrane, and once it goes there, we hope that this is still functional and start secreting the protein. So this is the whole overview of gene delivery in a nutshell. Obviously it has some advantages, you are not actually now delivering cell. So this is your own cell, and so immune system is not going to act on that, which is great. So this is autologous, or it is not really an implant. So in this case now your drug is gene instead of being cell. So we want to engineer some novel carriers that will facilitate this process and make it better. So we then go to polymers as we have been doing throughout this course. So mostly cationic polymers are used, and why cationic is because DNA itself is highly negatively charged. So as I said, the DNA is composed of nucleotides which are composed of phosphate, and this is several negative charge is present on each of those nucleotides, and so you can imagine a 100 kilo base pair DNA will have quite a lot of negative charge on its surface or on its structure, and so you can use cationic polymers, and that way these DNA molecule interact quite strongly ionically with your cationic polymer. So these then result in formation of complexes. So here is one DNA, then you let us say you take up a polymer which is positively charged and you mix them in the right ratio, or you vary the ratio, and at some ratio what will happen is they will start precipitating out and form these complexes or create packets or particles, and depending on the ratio you can change the size and all, and then further you can modify these polymers with various ligands to target a cell receptor or to target some other function. Then these particles are big enough that they can be taken up through endocytosis, and once they are taken up through endocytosis you rely on their escape from the endosome to, and then eventually release of this DNA. So, how does the release work? So, outside the cell, so let us say if this is a cell, you have cytoplasm and you have extracellular. So, the extracellular environment is typically low ionic concentration is present, but in the cytoplasm you have high ionic concentration. So, what that does mean? What does that mean? That means that since there is high ionic concentration, the dielectric of this particular cytoplasm is high, and because of that this ion based interaction, according to Coulomb's law, dielectric is in the denominator. Since it is high, it is going to decrease, and so that means that the bond is not going to be as strong and this can separate out. So, that is how when this particles will come out in the cytoplasm from the after the endosomal escape, they will cause release of the DNA, and then the DNA can go into the nucleus and transfect and start producing what you wanted. So, this is the whole concept of using polymers. There are a few polymers that are particularly good at this, and some of these are polylysine, polyethylenamine, chitosan, some polyaminoamine dendrimers. So, here just some examples that I am giving you. You can use other polymers also, it does not have to be cationic polymer, but it is just seen that cationic polymers are much better at this than any other type of the polymers. And so, you can have a big polymer such as let us say PLGA, put your gene inside, and when the PLGA degrades, this thing can come out, but you then run into the problem that they do not really have good encapsulation efficiency, not a whole lot of DNA is going inside your particles, and then the release is also just relying on degradation rather than triggering when it is out in the cytoplasm. So, that is one issue, and but then you can still use it. These can slowly release this gene. Once they go into the body, you can make various sizes as we discussed already with the PLGA particle. You can make matrices, you can make gels, and some of the polymers that are used is PLGA, PVP, etc. for all these applications. Okay, we will stop here and we will continue rest in the next class. Thank you.